Topic of discussion for today is glycosylation of the viral glycoproteins in mammalian N-linked and O-linked pathway. So we will cover how this viral glycoprotein use the glycosylation pathway that is available or that are present in mammalian cells, and we will cover two major kind of glycosylation, that is N-linked and O-linked pathway. So the panel A covers or provides the example which are the viruses that has the glycosylation on their envelope part. And so these are some of the viruses that are shown here. This one of the example is that double-stranded DNA, that is HSV, has the enveloped virus. So and that envelope virus contains the glycosylation or glycosylated carbohydrate structures that evade or that helps in evading the virus from being detected by the immune system of the host. The other examples are this dengue, influenza, HIV-1, hepatitis B. And there are some viruses that does not have enveloped and are not glycosylated, such as uh, some of them could be single-stranded DNA, other of them could be double-stranded DNA. And the third one is shown here. Here is a rotavirus, that is double-stranded RNA. Though it is a non-enveloped virus, however, uh, it has been seen that it used the same pathway that is used by the other enveloped viruses. So let us start with the panel B, and first we will start from the N-linked pathway. So on the left hand side is the N-linked pathway. So uh, in the N-linked pathway starts in the endoplasmic reticulum, as we have seen in our many of our previous videos how this N-linked pathway happens. So this mRNA will start translating into the lumen of ER, and where this nascent peptide will attach via oligosaccharide transferase. After that, the glucosidase 1 and 2 will trim off this two of this glucose manosidase or two of this glucose monosaccharide, and then it will be detected by calnexin calreticulum that helps in the proper folding. And thereafter, with the help of this alpha manosidase and other manosidase that are present in the Golgi, this uh, uh, folded glycoprotein structure will move into the cis part of the Golgi. At the same time, the manos, its carbohydrates structures, or this signal this signaling sugar component will now have 5 manos and 2 N-acetyl glucosamine. And after that, what happens is that this glycnec transferase 1 will transfer 3 of the manos into the first part of this into the first branch of this carbohydrate sugar of the mannose, as we can see here. uh, oh, it moves from here to here. So what happens in this case is that this glucose manosidase will trim off this glucose manosidase will trim off this extra glucose on this Golgi manosidase will trim off this extra mannose, and so that it will get a structure of around mannose 5 and acetyl glucosamine 1. And after that, this glycnec transferase 1 will transfer it to the medial Golgi, where what happens is that after the after the after the removal of other manos after the removal of other manos with the help of not just Golgi manosidase 1, but with the help of other manosidase, what happens is that these monocytes will now be exposed to the fucosyl transferase alpha manosidase 2 and fucosyl transferase, so that they will form a different combination of carbohydrate structures, as it is shown here. Some of them will form a complex structure and some of them will form a hybrid structure in the trans-Golgi region. And this decorated carbohydrate decorated glycoprotein, or we can say the viral glycoproteins in this case, will then be exposed to their extracellular matrix or in the cytosols wherever they are destined to, so that they can infect the cells and other cells within the they can infect within the cells and also infect other cells. The last part is the O-linked pathway that is mainly occurred in the Mucin type. So what happens in this case is that this GalNAc will attach to the serine thiorin part of the amino acid of the glycoprotein, and after that there are different enzymes that are present for O-linked that generally formed four kind of structure. First one is that core 1 structure, core 2 structure, core 3 structure, and core 4 structure. So these are the commonly exist O-linked structures are formed. However, they can be further modified, and these structures can be elongated depending on the kind of viruses that are infected uh, that are infected within the host. So this is a general overview how this glycosylation of the viral glycoprotein occurs in the mammalian cells, and we have covered two pathways, that is N-linked and O-linked pathways. If you need to understand these things in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.